Hello everyone. Assalamu alaikum. My name is Dr. Lakho, and I will be giving you an introduction to chapter cells as far as the syllabus 5090 of Cambridge Biology is concerned. This will be with respect to the revised syllabus for examination to be given in the year May-June 2023 onwards. So let's start. Okay, so as you can see, this page shows you a content overview about the list of the topics which all students who will be appearing for the Cambridge O-Level Biology 5090 exam for the year 2023 onwards, these are the topics that we will be covering as far as the syllabus is concerned. So just to give you a content overview. And then on this page, an assessment overview about the different kinds of papers and the different options that have been given for students who want to appear for this exam. So basically, they have to take 3 papers in total, so as to give them an aggregate of 100 marks. For that, either they have the choice to give a paper 1, a 2, and paper 4, or they could give paper 1, paper 2, or paper 3. All these examinations are externally assessed, and most of the schools, they give the option of ATP instead of the practical test these days. So that's more common. So of course, I'll be dealing with the paper 1, 2, and 4. As you can see, very quickly, I'll just give you an idea about paper 1 is a multiple choice paper of 40 marks, which comprises 30% of the weightage of the total paper, right? The total paper, let's say if we say it has 100% weightage, so out of that 30% weightage is for paper 1, where we have 44 option multiple choice questions externally assessed. And then we have paper 2, which is a 1 hour 45-minute long paper of 80 marks, comprising 50% of the weightage of biology paper. It comprises of short answer and structured questions, no essay questions. And last of all is the paper 4, which is the alternative to practical. In short, we also call it ATP, which comprises 20% of the weightage of the total paper, in which the questions that are asked are based on experimental skills in section 4. Most of this time, the students who are going to school, not in every school actually, but in some of the schools, the students are given exposure to different kinds of practical techniques. And based on the experience that they have on in dealing with the practical exposure, they are asked questions which are purely theoretical, but those theoretical questions are based on their experimental experience. But at the same time, all those students who do not go to schools or who do not have access to these experiments by performing them themselves, or maybe just, observing the teacher carrying out the experiment, they don't need to worry. It can be easily covered by showing you demonstrations and videos so that you get an understanding. So that will be covered also in my lectures. Moving on to the syllabus outline. Now, this syllabus, which you can see on the screen, is the new syllabus. Most of the things, not many things have been changed, but some chapters have been added, some revision has been made. So for that reason, I'll be sharing the new syllabus only with you. So it shows you cells, which is unit 1, and cells unit 1 itself has been broken down to unit 1.1 and 1.2, in which 1.1 deals with cell structure and function, and 1.2 deals with specialized cells, tissues, and organs. So today, the objectives or the learning outcomes that we will be covering are learning outcome number 1 and learning outcome number 2. Let me just read it out for you. Learning outcome number 1 means we'll be examining under microscope animal cells and plant cells from any suitable locally available material using an appropriate temporary staining technique such as methylene blue or iodine solution. And the second learning outcome is that we should be able to draw diagrams to represent observations of animal and plant cells examined above. So let's start with this. Of course, since this is just a theory-based lecture, so I'll try my best to give you an experience of actually doing the experiment yourself, or at least, if not doing, getting an idea how could this be done in the real world scenario. So let's start. This is an optional slide which actually gives you information about how teachers plan out activities. So I will be keeping this slide as a part of my presentation or a part of my notes. So whoever wants to consult them are more than welcome to do so that they get an in-depth understanding of how we go about as far as the syllabus is concerned, how much depth is required, how is the syllabus meant to be covered. It's an extra information. Those who are interested can always go through it. Now, starting with the concept of cells. Almost every student who opts for O-level biology has got some basic understanding, fundamental understanding about what cells are. So basically, a cell, I would define, is the smallest structural and functional unit of life. Since biology is the subject which is concerned with the study of life, so the smallest structure and the smallest functional unit of every living thing, be it a small bacterium or be it as big as a human being, is composed of cell. The tiniest part of our body and the tiniest part of the body of a bacterium is a cell. And it has got all the capabilities, all the features of living independently on their own, growing, respiring, moving from one place to another, and doing many other activities. But our human eye cannot see those cells because they're extremely small, right? So for that, of course, we have to take help from microscopes. For example, you can see in front of you two kinds of microscopes. One of them is a very simple microscope like this, which has a lens at the top. It has some lenses over here as well. It has a mirror over here. And most probably what we do is that if you want to observe something very small, let's say a cheek cell or a plant cell or any other animal cell, we put that cell or that tiny specimen on a slide and we keep the slide over here and we try to look at it by using this microscope. So I'm sure most of you must have had the experience of looking at this microscope in pictures, and some of you might have had the chance to look at it in real life also in schools. So such kind of a microscope is known as a light microscope. The reason why we call it a light microscope is that it uses the visible light, right? The visible light, the sunlight or the tube light, in order to view any small object under magnification, right? And we use different kinds of lenses depending upon their magnification. Let's say the lens which I have used over here is giving you a magnification of 10 times. And this lens which I'm using over here is giving you a magnification of 40 times. So 10 into 40 will be equal to 400 times magnification. So in that case, I can see any object by magnifying it 400 times. So this kind of a simple microscope can enable me to see objects which I cannot see with my natural eye. And let me tell you, for your information, that a good quality, powerful light microscope can magnify things to the maximum of 1500 times. So we can have a look at different kinds of plant cells and animal cells through this light microscope. If something is extremely tiny, which cannot be seen even after magnifying it for so many 100 times, 1500 times, we can use an electron microscope, which is of course not easily available. It is found in big research institutes. So an electron microscope can give you a magnification of 250,000 times. So if you want to look at very tiny parts of a cell itself is very small, but if you want to further zoom into the tiniest parts of the cell, let's say if you want to see what is inside the nucleus, right? If you want to see any other small structure of the cell, you can do that by using an electron microscope. So this means electron microscope will enable you to get a far more greater detail as compared to light microscope. Even before the invent of light microscope, the way by means of which people could see objects by magnifying them was the simple use of a magnifying lens, which could magnify roughly around 10 times. So cells can often be like the cells, the living cells can be seen just as big as dots with the magnifying lens. But of course, we don't just want to see dots, we want to actually see what's inside that dot. So if you want to see a cell with a magnifying lens only, we will be only able to see like this. If we want to further magnify it, we can use a light microscope so that we are able to see these many details. And if you want to further zoom into the details, then we can move on to the electron microscope. Okay, this was some detail about how to visualize cells by using different kinds of techniques, either a hand lens or a light microscope or an electron microscope. Let's move on to how can we actually prepare specimens so that we can visualize them under a microscope. So just to give you an idea, let's say if the specimen that we are using is a stem, right? If I want to see that a plant stem, let's say this is a plant stem, right? These are the roots, the leaves, and this is the plant stem. If I want to see what kind of cells make up a plant stem, so since the stem is quite thick, right? I would want to take a very thinnest possible slice or a section of the cell. So I can take a slice, or I can cut it in various directions. I can cut the stem like this, okay? Or I can cut the stem in this direction. Now let me just draw it bigger to show you how I can cut it. Let's say if this is the stem, right? Now I can, and if I have a razor, okay, I have a thin blade or a razor, I can use this razor to cut the stem in this direction across. So if I can cut the stem in this direction, okay, I'll get a very thin slice. Since the stem is cylindrical, so I'll get a thin slice of a stem like this, which will show like this. And I can pick up this slice, keep it on a slide like this. For example, this is the microscope slide. I'm going to transfer it like this. And I'm going to lift it up and keep it on the microscope over here. Let me draw it. Let me just zoom into it. And now you can see this is my slide. And this is the section of the stem that I have taken, the thinnest possible section. Okay. And now I'm going to keep this slide with the thinnest possible section of the stem, which in this case is a cross section. I keep it. I use this lens and I use these lenses to visualize it under magnification. And I'll be able to see actually what makes up the cells of a stem. This is a cross section. I can take a cross section also, and I can take a longitudinal section also. Longitudinal section would be, let's say if this is your stem, I can cut it like this, okay, and take a thin slice, or I can cut it in this direction and take a thin slice, right? And again, I can place that thin slice and observe how the stem looks like under high magnification with a light microscope as far as its longitudinal section is concerned or a cross section is concerned. Another name for the cross section is a transverse section, right? So let me give you an idea about how a stem would look like under a cross section and under a longitudinal section. So if we look at a stem under a cross section, it looks like this. And if we want to have a look about how a stem looks like under a longitudinal section, it looks like this. So this is longitudinal section. And these two are the cross sections, right? So the structures are the same. It's just the angle which is different. So why am I focusing on these different kinds of sections? Because you will be given, lots of opportunities to visualize different kinds of structures in their transverse sections and in their longitudinal section forms. So you should be aware of such terminologies. Now I would be sharing some pictures of real human cells or animal cells and real plant cells. So right in front of you, you can see a light micrograph. Any picture, like for example, if I am visualizing any cell under a light microscope, and if I have a camera fitted in the light microscope, or I can simply use my camera of my cell phone only to take a picture of what I can see through the lens of the light microscope, then that kind of a picture is known as a light micrograph. So right now, which you can see in front of you is, this is also a light micrograph. And this is also a light micrograph of an animal cell. And this animal cell is a cheek cell. You can see in the diagram over here how a toothpick is being inserted into inside the cheek, in the inner part of the cheek of this man, and this toothpick will be smeared onto the slide, and then that slide will be, of course, full of those cheek cells. And if we put that slide under a microscope, we will be able to see lots and lots of these cheek cells. Now, to tell you one very important thing, remember this, guys, that our cells are not colored. They don't have any color at all. The reason why we can see them as blue colored or pink color is that various stains or dyes have been used to color them so that they become more obvious and we can see them clearly and we can identify different parts. So there are different kinds of stains which are used to color such cells. For example, the most commonly used stain are the iodine, right, and methylene blue. Iodine is usually brownish or reddish in color, and methylene blue is, of course, blue in color. So that's why we use these stains so as to make the structures obvious. So this is how a cheek cell looks like, okay? And this is the real cheek cell. Now, if I ask you, and sometimes examiners do ask you to look at this drawing, the picture, and to make a drawing. So I can roughly make a drawing like this, in which I have drawn the outer boundary and the nucleus, and the cytoplasm contains many granules. It contains some small vacuoles. I can also see the nuclear envelope, so I have labeled that as well. So roughly, this is the way by means of which we look at the real light micrographs and we can draw diagrams out of it. Let me show you another slide which shows you details about different kinds of stains which are used. So the stain which is preferably used to stain plant cells is the iodine stain, which is used to stain cells like onion, right? And methylene blue is used to stain animal cells, right? This stain does not immediately kill the specimen, so it is ideal to observe the animal cells. And iodine is actually ideal for staining the plant cells. And it actually makes it possible for an observer to see the structures of a cell easier under the microscope, because to see something which is black and white and to identify different structures is difficult. Another picture which shows you 3 different diagrams. One diagram on the extreme right, which shows you cells which are animal cells that have been taken from a trachea of a mammal as seen through a light microscope. And then you can see this structure which has been taken from a plant cell. And we see lots of these green structures because plants can photosynthesize, they can make food. So since plants are green in color, that's why they have, since they are green in color due to the presence of chlorophyll, so we can see these chlorophyll or green coloring substances in many plant cells, right? And on the extreme left-hand side, we can see a drawing. That's not a real picture, but that's a drawing of a plant cell. Acha, in the old textbooks of biology, the word a typical animal cell and a typical plant cell was always used. So nowadays, these terminologies are no longer used. Why? Because there is it's such a huge variety of animal and plant cells that there's no such thing as a typical plant or a typical animal cells. You can have a wide array, a wide variety of features within the plant cells themselves, and the same thing is true for animal cells. But yes, there are certain features which are very typical for animal cells, and there are some features which are distinctive for plant cells, and we'll be doing them. In the end, I'll be drawing two diagrams with my own hand, one of the animal cell and the other of the plant cell. Usually students think that the animal cells are circle in shape and the plant cells are squarish in shape. Guys, this concept is totally wrong. Animal cells or plant cells can be of any shape. What makes them distinct as animal cells and plant cells are their distinctive features. So let me give you a very brief detail of their distinctive features. So let me draw an animal cell. Every cell, whether it's an animal cell or a plant cell, has a boundary or an outer boundary, which is known as the cell membrane. It's a living structure. Every living cell has a structure inside the cell, which is known as the nucleus. To state briefly, because I will not go into the details of nucleus today, I would say this is that structure within a living cell that controls all the activities of a cell. The space between the nucleus and the outer membrane or the cell membrane is filled by a jelly-like substance, which is known as the cytoplasm. Cytoplasm is that part of the cell where all chemical reactions take place. We can make an analogy of the cytoplasm with a chemical laboratory. Just like we carry out all different kinds of reactions and experiments in a chemical laboratory, the cytoplasm is the place where all different chemical reactions take place inside the cell. Nucleus controls all the activities of a cell. Cytoplasm is the place where various chemical reactions take place. Cell membrane is the outer boundary or the gatekeeper of the cell. It controls the entry and the exit of substances into and out of the cell so that the cell is protected from all kinds of dangerous substances. Then, if I compare this to a plant cell, let's say I'm drawing any shape of a plant cell, this shape. So the plant cell also has a cell membrane. Just like an animal cell, it also has an outer boundary or a cell membrane. The plant cell also has a nucleus, but now comes the difference. The nucleus in the plant cell does not lie in the center. It lies a little bit to the edge of the cell. The plant cell also has cytoplasm just like the animal cell. So this means the space between, all the space between the nucleus and the cell membrane is the cytoplasm. Now comes an additional feature of the plant cell, which is not found in the animal cell, which is the cell wall. It has this outer boundary or the cell wall. A cell wall is a very rigid, tough structure, a dead structure. It doesn't have life. That lies outside the cell membrane. It's a rigid, tough, dead structure. A plant cell also has another important feature, and that is it has a huge, this much big, the size may vary, structure, which is known as the vacuole. A vacuole is that part of a plant cell which stores essential substances, right? It can store carbohydrates, it can store water, it can store different kinds of pigments, it can store mineral salts, various substances. Animal cells also have vacuoles, but the vacuoles in the animal cells are small, they are numerous, and they are temporary. So, vacuoles in the animal cells, small, numerous, and temporary. Whereas the vacuole in the plant cell is one big vacuole. It is permanent and it is large. So these are the very, very basic and fundamental differences between plant and animal cells. The details about different other structures of the plant and animal cells will be covered in the upcoming lesson. So bye everybody.